You want to copy? Copy this one?
Yes, one
Yes.
file election.
Where I should copy? Where you want me to paste? This one,
Yes,
huh?
yeah, from the science. PPT is there, sir. No, no, not.
How to paste it?
Yeah, yeah.
College of Pharmacy, completed M.Pharm in Pharmaceutics from Savitribai Phule Pune University and PhD from JNTU. Sir is having total 21 years of experience in the academics. Moving towards research profile, Sir is having near about 170 research publications in national and international journals of repute with good citation index of 957 with NCT index of 17 and I-10 index of 33. Moving towards his achievements, Sir has published nearly 20 books of national and international repute, 15 book chapters, funded research projects 3, industrial product development 2, national and international awards. He is a recipient of near about more than 12 awards at national and international level. Sir is guided near about 60 PG students and 2 PhD students, and currently 6 PhD students are pursuing under PhD under him. Welcome, sir, on behalf of GS MHG Copper. Now, again, it's my immense pleasure to introduce our post-lunch session, that is session 3, speaker, Dr. Jaikant Chandrasekharan, sir, Assistant Professor in Pharmacology at SPTM NMIMS University. Sir completed his PhD in Biochemical Pharmacology from Pondicherry University, did M.Pharm from Sastra University and B.Pharm from PHG College of Pharmacy. Sir is also having 11 years of academics and research experience. Sir currently working as Assistant Professor at SVKM's NMIMS University, Hyderabad, Telangana. He is before that Associate Professor at PHG College of Pharmacy, Coimbatore. Sir attended various national and international conferences and attended 6 FDPs. Moving towards his research profile, Sir is on his credit 1 patent and international publications of 11 publications will be there, and citation Google Scholar citations of 112, H index of 5, I-10 index 2, and Sir has guided M.Pharm students in pharmacology, 9 students. And moving towards his achievements, he is All India rank 1 in entrance for doctoral program of Pondicherry University. Sir as a recipient of Senior Research Fellowship by CSIR India, and he has also qualified GPAT in 2011. I welcome you, sir, on this auspicious occasion of international conference on behalf of GS Sir Dr. M.S. Gosavi College of Pharmacy. Please, sir. Thank you.
Thank you, sir. I would like to request Dr. S.V. Amrutkar, sir, to felicitate our today's session chair, Dr. Sanjay Kshirsagar, sir, with the bouquet and shawl. Please, sir. Thank you, sir. I would like to request Dr. S.V. Amrutkar, sir, to please felicitate our resource person, Dr. Jaikant Chandrasekharan, sir, with the bouquet and shawl. Please, sir.
Thank you, sir. Now I request Dr. Jaikant Chandrasekharan, sir, to please begin with the session.
Okay. Good afternoon, everyone, and thank you for having me here. Am I audible? Yes, fine. So today I will. Yes. Yeah. Yeah, fine. Yeah. Today I'll share about the journey, some success stories, some failure stories, and what I am currently doing. And I'll also nurture who are passionate in research. I'll show you how to design a research protocol. Okay, so we'll see about. So I have made a title like well thought out and sought out strategies in drug discovery in this omics era, because this is a omics era. We have humongous data. We have genomics, we have transcriptomics, we have proteomics, we have phosphoproteomics, we have glycomics. So the data are so humongous. We don't know where to look at. How to synthesize all those data in a meaningful way. So what I am going to do today is I'll show you how an industry, a typical industry, will look into a drug discovery. We used to read in a book, we used to follow some slides, but how an industry will perform a drug discovery with a case example and also my experience with that. So why to study and how to study? These are the two important questions which you need to ask to yourself whenever anything is put forth in front of you. That is the only thing which will kindle your mind. Why to study? Why to study this pharmacology? Why to study the medicinal chemistry? Why to study this topic? So for that I'm going to give the answer. So as I said, there are humongous data which are in a it's in a chaos form, so we have to make it in a meaningful way. So for that we need to know little bit about the history, how the chemical become a medicine, little bit history we have to look back, then only we can have a very good idea about the present. So we know that drug discovery is a huge attrition process, like what is happening in today's pharmacy college, huge attrition in faculty, especially pharmacology faculties are just coming and moving. Likewise, this drug discovery is also a huge attrition process, and it is a putative transmogrification process, which means that multiple departments have to work hand in hand to make the product a successful one. And this pre-marketing phase is called as drug cycle, and we are knowingly or unknowingly, we all are codified or embedded in ICH guidelines. So ICH is, we know our pharmaceutics, you will study ICH like, okay, something like that, but it's a scientific consensus of the expert from different domains. So if you are very clear with ICH, you are an expert. So when you speak about drug discovery, you can't skip out of ICH. Toxicology means it has to be done in GLP certified laboratory. Otherwise you can't submit for regulatory submission. So likewise, everything is codified, and regulatory affairs department will also work together, and integrative approach of all this discipline give an eventual success. So in morning, the earlier talk which is given by madam have given little bit of platform, but I'll cherish the platform much more. How the medicines are discovered is a fundamental question. How the medicines are discovered. So from indigenous transfer of knowledge, from the treaties we have, from the traditional system we have, we are discovering, and by human experience or discovery by chance, which is called as serendipity. So discovery by chance, like how the penicillins are discovered. And nowadays the complexity is arriving because we are speaking about targeted medicines and their importance. Because of this more technological and biological breakthrough, we have high-end mass spectroscopy where I can, within 4 hours, I can come up with 600 differentially regulated proteins. So we are trying to identify the ingrained roots of pathology more deeper and deeper, so that we can precisely identify this molecule is a regulator for a disease. This is overexpressed, or this is less expressed, or so this technological and biological breakthrough made today the medicine director to identify the pathological ingrained roots of the pathological process, so that we can target the self-centric or targeted medicine can be applicable. So how it makes the system more complex? We know, we study G protein coupled receptor. That is a slimmed one, sorry. That is a slimmed one. Okay. we have nearly 3,000 to 4,000 molecules operating inside a pathway, and we just joined the important nodes, and we will give a slimmed one for your general understanding. And this is a keg pathway. This is also a slimmed one. Yes, this is also a slimmed one. So out of nearly 4,000 to 5,000 regulator, we just slimmed something and have shown. So if I want to study about a particular, for example, here I want to study about a CHEK1. It's a new target for cancer. It's an emerging target for cancer. You can see the CHEK1 is here, and it is interlinked by so much of pathways. So I need to understand how to look at the pathway and how to study all these things. So the science becoming complex day by day because of this biological breakthrough. So when we speak about the history again, we know that discovery into IND filing, it will take 1 to 5 years. IND to NDA, it takes 6 years. Review and approval, it will take 1.1 years. Total billion of 1.3 billion will be spent. So what is the success rate of spending so much of a year and money to it? So you can see only 1 in 1,000 compounds will reach the market. So candidate for a new drug to treat a disease might start from 5,000 to 10,000. That all I'll show in the coming slide, how the industry will apply. Before that, we'll see little bit of history. So from 5,000 to 10,000, we will scrutinize 250 in the first year. Then from the 10 will get into the clinical stage, and 1 will be claimed as a drug, or it goes to the phase 4 clinical trial, that is post marketing. So we are getting into the topic now only. So we need to know what actually we are doing in research. We will develop a hypothesis, and we try to justify that, or we try to validate the hypothesis from low validation state to higher validation state. What is the meaning of lower validation state is, see, I have derived a hypothesis, this drug can stimulate insulin. What I will do, I will put this drug in a beta cells, that is in vitro study, and I will try to make my hypothesis right or wrong. If it is work, then I will go to the in vivo, in animal model I will check, which means from low validation I am going to the next level of higher validation. From animal I will move to the clinical, that is phase 1, that is with human. So in medicine we used to say, do no harm, whether you are treating or not, that is different. The drug need to be very safe. So for that only this validation is done. So this you need to know where you are going to work, like where you have to start, in vitro or in vivo or clinical thing. So you have to define your ground zero first. And when we compress the drug discovery process in a small thing, first is a target identification. So you need to identify the target. It can be a first in class target, or it can be a best in class targets. So these are the keywords which you need to know when you are searching for the information. First in class means for the first time this target was exposed and it was shown related with the disease. That is called as first in class. Or the rest are best in class. The target is already validated, and most of the pharma companies will work on the best in class targets. So target identification, all this genomics, proteomics, transcriptomics, we try to identify which is driving the disease. That is the target identification. That itself is a PhD or a postdoc in abroad. It will take 1 to 3 years. And then after target identification, we generate lead, that is a lead compound for the target by using different techniques. That will take 1 to 2 years. Then after that, we further modify the lead to make it pharmacokinetically more portable. And then we'll go to preclinical and formal development in the clinical phase. So totally it will take 12 years. Now because of this technological advancement, you can able to generate the lead in 7 months, and lead optimization can be done in 9 months. Preclinical development can be done in 1 year, and formal development, that is the clinical phase, phase 1, phase 2, phase 3 can be done in 4 years. So cumulatively we can generate a good clinical drug or a candidate within 7 years. So these are the primary important things. Now let's get into what is the meaning of a target or a compound selection. or a target choice. So, as a pharmacologist, if you ask me, then I'll say my drug has to go and inhibit the target and it has to produce a response. If you ask a chemist, a medicinal chemist, he will say that molecule need to be given orally. So it should have a very good pharmacokinetic properties. So the integration of this biologist and the chemist need to be there if you want to select a good compound, because the integration of both the chemistry as well as the biological discipline is important for selection of a good. So now this is a very, very important slide, how the industry operates. If you know this, you know where to start and where to end. So once if I want for example, if I want to develop a drug, so first focus is whether I am going to develop the drug is a first in class or best in class, whether any drug is available for that target. If it is available, what are the limitations of those available drugs and how I am going to overcome that limitation. That is called as best in class. First in class means I am the first person who is going to develop a drug for the target. That is called as first in class. So you need to know whether you are going to develop a drug for first in class or best in class. And another important thing is 70% of the pharma companies work on best in class, which is already known targets they work. And the second thing is, I want to make sure whatever compound I am going to generate need to be structurally unique. Then only I can go for patent. I can commercialize the product as well. That will give me money. Okay. And in terms of solid pharmacology, what are the key things? The potency that meets or exceeds gold standard, which means the already available drug. It need to be equivalent or more than that, the potency in terms of potency. And the second thing is target selectivity. Whenever we say about a target, for example, I am going to speak about DPP4 inhibitor, the anti-diabetic agents. They come from one family. They have close association. They have brothers and sisters. They are called as homologous targets. We are analogous targets. We need to check what are those targets which has a similar active sites, so that our drug will go and bind only with the DPP4, not with any of the family members. That selectivity need to be more than 1000-fold. This is very, very important. Why most of the time we will fail is the selectivity won't be there. So it will produce a lot of toxicity. And it should possess excellent drug metabolism and pharmacokinetic properties, which means that no drug-drug interactions and suitable for oral dosing, limited metabolism, and robust efficacy in rodent immunity. And here I have shown something called as first tier screening. What are the things? Is it visible? I don't think so. So once I find a chemical, first I have to go for an enzyme binding assay or a receptor binding assay in vitro, and it has to produce the EC50, that is effective concentration, or IC50, which need to be less than 10 nanomolar. Then only you can able to translate that chemical into a drug. Next criteria, and solubility need to be 0.1 to 0.2 milligram per ml. Whatever compound you are going to get, it has to get solubilized in 0.2 to 0.2 milligram per ml in pH of 7. This is a body pH. Then only it can get translated. And then that mouse liver microsomal enzyme, human microsomal enzyme, rat microsomal enzyme. So if we put the drug, the T-half need to be greater than 60 minutes. Nobody will teach this. This is how the industry will operate. So these are the important things where we will fail. And hERG binding, this is very important. This is a potassium channel. So if it blocks, then this will manifest Raynaud points or the heart failure. So the hERG binding should be seen, and have to check with 5 major cytochrome P450 enzymes, and we say that it has to get minimal metabolized with this one. And mini Ames test, I hope you all read about Ames test. It will show mutagenicity. Okay, so the compound have to show 10 nanomolar EC50. Then it has to get solubilized in 0.1 to 0.2 milligram in ml at pH 7, and it has a half-life of greater than 60 in mouse, human and rat microsomal enzyme, and HERC blocking should not be there, and it should not get metabolized by 5 major, otherwise it will become a substrate for the cytochrome P450 enzyme complex, and for many aimsters it need to be negative, otherwise it will be a carcinogenic agent. So if we filter, then out of 10,000 compounds we will get some 1,000. So second level of filtering is also there, that is the second tier of studies. So at least when you plan for a PhD, at least you have to do the first tier level and make sure it can get translated to the next PhD candidate who is going to work. So this is how the industry will operate, and I am not going in depth about the second tier and third tier. Yeah. Yes. So how an industry will develop a lead, how an industry will develop a chemical. So what are the techniques they will adapt? So this is a cycle I have shown here. So you can see they will screen for fragments or shape, which is called as a structure based, and scaffold morphing. If one scaffold is producing an activity, then they will try to morph a similar kind of scaffold from the visual screening, just randomly screening all the chemicals available in the database, or de novo design, BIOS library, any known inhibitors, any clinical trial inhibitors which is known. So we can able to generate, these are the features. It has to possess 2 donor groups, 1 acceptor group, like that we will make a feature. So for example, if I want to make a Xerox copy like you, then the hand need to be like this, leg need to be like this, height need to be, that is called as pharmacophore, like that we used to design, and we try to screen. And likewise, we try to do that, and from that, as I said, we do biochemical or cell based assay, from which we will try to identify IC50 or EC50, and that EC50 need to be less than or equal to 10 nanomolar, then only you can translate. So once it is done, we need to find out how this drug is binding with the target protein. That will be done by X-ray crystallography. Now cryo-EM, all this have come, so we can get within 4 hours how it is binding, what is the nature of binding, what kind of interaction it makes, so that you can able to further design or define or refine the chemical which you have identified. So this process will keep on repeating unless you will get a very potent molecule with less side effect and very good ADME properties. So likewise, this cycle you need to understand. Now let's get into a classical, you all known case study, that is DPP4 inhibitor, how the allogliptin came into the picture, how they have identified the allogliptin. I am going to tell a story how it came into the picture. For that you need to know little bit. So most of the idea will come from the bedside, that is from the physician. They blindly say this is what happening, and that problem will come to us. We will think about it. Likewise, what is the problem is, the clinician found when they give oral glucose to a person, so when oral glucose is given, this will releases glucagon like peptide 1. Okay, and this release is not happening when they give the same glucose in IV. Got it? When a person takes glucose orally, this glucagon like peptide 1 is released. When the same glucose is given in IV, this glucagon like peptide 1 is not released. And they found out what is actually happening. What is the role of glucagon like peptide 1? So what is the role of glucagon like peptide 1 is, we know glucose is a stimulator of insulin. How it is stimulating is, it is stimulating the insulin by increasing the level of glucagon like peptide 1. And this glucagon like peptide 1 is metabolized by the enzyme dipeptidase, that is DPP4, this enzyme. So if I want to increase the half-life of GLP-1, if I inhibit this enzyme, then we can increase the half-life of GLP-1, and this will enhance the activity, or it will produce anti-diabetic activity. So that is how. So how this crux or how this concept came into the picture is when they give oral, as a physician they are looking, we give oral insulin, this glucagon like peptide 1 is increased. When we give IV, this is not increasing. What is the problem? When we look into it, they found out this GLP-1 is a key regulator for insulin release. But its half-life is 1 to 2 minutes. If I inhibit the enzyme which is degrading the glucagon like peptide 1, then that could be a good strategy. That is how this allogliptin came into the picture. So now I want to develop a drug for DPP4. So before anybody who want to do a research, you have to make something called as absolute criteria and relative criteria. So what is absolute criteria is, I want a highly selective and very potent DPP4 inhibitor. Highly selective and very potent DPP4 inhibitor, and that inhibitor should not have no cytochrome P450 interaction. Second, and it need to be given in a once daily dosing. All are having commercial value. Okay, so if a person is taking once daily tablet, then it is good. I can commercialize the product very well, and it's easy. And it need to be orally active. And if it is a best in class drug, it need to possess superior efficacy and safety profile. When all other product, for example, I am speaking about allogliptin, vidagliptin is also under DPP4, it need to be superior than vidagliptin. And what are the relative criteria? These are the absolute criteria. If the absolute criteria is matched, I am the millionaire. If it is not matched, what I have to look at? I should always keep a relative criteria. Relative criteria is, it need to be, the drug which I am going to identify need to be equivalent or superior to the best known competitor, that is DPP4 inhibitors. What are the best known competitors are from Novartis and Merck. So when compared to these two competitors, at least it need to be equivalent or superior. These are the criteria which we need to keep. And as I said, once you identify a drug which is having a nanomolar concentration, we need to go for X-ray crystallography. And after doing X-ray crystallography, you can able to identify how it is interacting. These are the pictures in the left hand side. How it is interacting, which part is interacting, which is producing hydrogen bond, all these things will give you an idea. And here is allogliptin, the molecule which the company have identified. This sitagliptin, vidagliptin and sahagliptin are the other molecules, and they look into the structure, whether the structure of allogliptin is similar or different. Yes, it is unique. Allogliptin, you can see it is different from all other structures. So I can eventually go for patent. Got it? So this is how the fundamental criteria. And as I said, where we always look into the things. DPP4 means DPP4 only we will see. We should always look into their homologs which have the similar domain or active sites, like FAP alpha, DPP2 is there, DPP8, DPP9. So these are the homologs, and whatever drug I have identified need to bind with these homologs in a very very less level, or it should not bind. And the fold of binding should be greater than 10,000 fold or 1,000 fold, as I said earlier. So that is how you should. And also you should always check into the homologs what is activity, whether it has any essential or important cellular activity like T cell apoptosis, because whatever drug you find, and if it is inhibiting these homologs which has the integral activity with day to day life, then automatically your life will be compromised. It will be acting as a poisonous agent. So how they have started this experiment? So here are the drug allogliptin, and this is DPP4. You can see that allogliptin is inhibiting at 6.9 nanomolar, whereas galvesmet, that is vidagliptin, 23.8 nanomolar, and ganavia, that is 12.1. These are the other two competitors. So the first criteria is this allogliptin is having a very good potency. And next is, I am doing an enzyme binding assay, how far it is selective for DPP2, DPP8, DPP9, FAP, PERP, all these things. You can see for allogliptin, the selectivity is greater than 1 lakh. The 1 lakh fold, we have to give so much of concentration of the drug, then only it can go and inhibit DPP2, DPP8. Whereas Vidagliptin, you can see DPP8, it has 1400. So here it gives some idea that whatever compound you design, it need to be more selective to the target, not to the homolog. So that is what we are identifying here. So I have did an enzyme binding assay and I found out the two criteria what I kept is it has to bind with the target at nanomolar concentration, and it has more selective to the DPP4, not with any of the homologs. Now it is done. So next criteria is I have to validate. Now this DPP4 is inhibited. If it is inhibited, then GLP-1 level need to be high. Yes or no? Are you able to follow what is the concept here? So alogliptin, what they do is, since it is a anti-diabetic drug, here we need to select a valid animal model. You can't select, since I have streptozotocin, I can do an anti-diabetic activity with streptozotocin. So what it replicates? Always the pharmacology will ask, a pharmacologist will ask, streptozotocin induced diabetes is a type 1 or type 2? It is not type 1 and type 2. Not at all. So when we look into the functionally, so it will fall under a category of non-obese diabetic patient. A patient who is diabetic, he is non-obese. It will reflect that status only if you use streptozotocin. So in non-obese diabetic patient, whether my drug is working or not, that is the first criteria. For that we are giving streptozotocin and we are giving alogliptin, and we try to find out whether it is inhibiting and increasing the GLP activity. So you can see that yes, it is inhibiting DPP4 activity and increasing the GLP-1 activity. So the first claim I have put here is, so my drug will work for non-obese diabetic patient. Right. Now there are obese diabetic patients. There are something called as pre-diabetic or impaired glucose tolerance patients. So for that, what model I should go? That I have to think next. And first we are inhibiting DPP4 and GLP-1 is increased. If GLP-1 is increased, obviously glucose level has to get increased. That I need to study, no? So for that OGDT test we are doing, and we found out that yes, because of this inhibition, the plasma insulin level is increased and glucose level is getting decreased also. So that one line concept, how it is working. If GLP is increased, insulin will be produced high and it will reduce glucose. These are the key parameters which you need to measure. No need to do all antioxidant, because I used to see students, since we have antioxidant kit in our laboratory, they used to do all antioxidant assay. ROS, not required. What is needed, which is a correct endpoint, that is enough to prove. And anyone say what is the difference between anti-hyperglycemic agent and hypoglycemic agent here? Both are same. Anti-hyperglycemic agent and hypoglycemic agent, both are same? No. Insulin is an anti-hyperglycemic agent or hypoglycemic agent? Metformin is an hypoglycemic agent or anti-hyperglycemic agent? Why? See, if insulin is injected to a normogenic, that is normal person, it will reduce the glucose level of the normal person also. Those agents are called as hypoglycemic. If a normal individual taking metformin, it won't reduce the blood glucose level. They are called as adaptogenic drugs. That's why using the terminology is also very important. Which drug we need to call it as an hypoglycemic and which we need to call it as an anti-hyperglycemic agent. Right. So for that, what they did is, what my drug is doing with the fasting glucose level, whether it is changing or not. See, you need to be very clear what are the things I should measure and how I need to correlate. So fasting blood glucose level, they are studying in a normal rat. Normal rat, they are giving the drug and they are trying to see whether it is reducing the glucose or not. If it is not reducing the glucose level, then we can very well call it is an adaptogenic drug and it is an anti-hyperglycemic agent. So that is also they are making. So this is how we should know. We should first understand the pathology and then we have to make the endpoint. So now it's very clear the allogliptin is not a normogenic one, it's an adaptogenic one, and it can be used for non-obese diabetic patients. Now the question comes, what we can do with the patients who are obese, who have insulin resistance? So for that, is there any model is available? Yes, we have DB-DB model. Okay, so DB-DB model is, it has a leptin receptor mutation, and it shows obesity, insulin resistant, and what are the features? Obesity and insulin resistant and type 2 diabetes mellitus. Yeah.
Please maintain silence.
Yeah, there are few listening to it. So this DB-DB mice we can very well correlate to the obese diabetic patients. So for that, this again, if I want to make a claim my drug is going to work on the obese diabetic patient, I have to do it on DB-DB mice, and I shows that yes, it is working on obese diabetic patients also. So for that DB-DB mice. So, and in the same DB-DB mice we have to confer how far DPP4 activity, GLP-1 level is increased, same thing. And I need to do whether it can be given for pre-diabetic patients or glucose or impaired glucose tolerance, who won't be having fasting blood glucose level of more than 200, but their range will be 180 to 200. They are called as impaired glucose tolerance. It can be managed by doing exercise. Whether this can be given for impaired glucose tolerant patient or not. For that, is there any model is available? For that, OB mice is there. So what it will do is, so it will display progressive food intake, insulin resistance will be produced, and moderately increase the blood glucose level. These are the features of pre-diabetic individuals. So they will have insulin resistance, they will take food excessively, but there will be a moderate increase, not more than 200 in the postprandial. So if I do in OB mice, I can make a claim it can be also given for the pre-diabetic individual. So this is how to select a valid animal model. It's not like what is available. What I want, for that what I want to do. If you want to work on ceftriaxone induced, then you should make a claim it is going to work for non-obese diabetic patients. Okay, so like that you need to develop a valid animal model for anything. And yes, and since insulin and glucagon are having an axis of regulation, if insulin is increased, glucagon has to get decreased, then only the axis is regulated in a good way, because it's an hormonal, the second delayed process, metabolic process. So what again we need to check the axis is not disturbed or not. If the insulin is increasing, the glucagon level is decreasing. When the glucagon level is increasing, proportionately the insulin level has to decrease. So this is also the another important endpoint. It is regulating the insulin glucagon axis. That what you have to say. And whenever we speak about the endpoints, these are the pharmacological endpoints, whatever I taught. There is something called as pathological endpoint. That is, in islets, whether it helps in regeneration, whether the insulin level is high or not. That's very important. So after treating, they found that the number of regeneration of these beta cells is very high, and the islets also have lot of insulin in it. So it helps in the regeneration of beta cells. So I make a claim now. There are 5 claims I can make. One is, my drug will act for non-obese diabetic patient. My drug will act on obese diabetic patient. My drug will act on pre-diabetic patient. And my drug will regenerate and protect the beta cells from degeneration. So this is how, when we go for a patent or when you are going to make a claim, you need to be very clear. This is how the industry operates, and these are the presentations from the industry. So I don't want to, this is what I want to discuss for efficacy point of view. And now I'll tell you few of the projects which I went into, and now they are in the phase 3 clinical trial. So I am fortunate that during my PhD I was mentored by Dr. K.M. Cherian. He is a Padma Shri and he is a renowned cardiothoracic surgeon. He did India's first open heart surgery, heart and lung transplant. All credit goes to him. He is a great mentor for us. So this is an international center for cardiothoracic and vascular diseases, where we will work with the stem cells. Have you heard of it? Stem cells. Yeah. So what we thought is, when I was doing PhD, my HOD, and what we thought is, so axonal regeneration, is it possible with stem cell or not? So that is, which means the peripheral nerve injury or axonal injury can be treated with umbilical cord derived Vatanjali. This is a topic. This we submitted, with this I presented in San Francisco, California, in transfusion medicine. So why we chose is an important thing. Why axonal nerve injury, and what are its bedside complications? Why we chose biological nerve graft? I will explain. Why umbilical cord, study design and outcome, translational value and limitation. All these things I am going to discuss, because before doing any experiment we need to know the logic, why we are doing it. Otherwise it's very, very difficult. So the first question is, why axonal nerve injury and bedside complication? When we talk with the surgeon or doctor, so surgery with allografts is the only treatment option. If anybody got injured with the axon or the nerve got damaged, then the only option is an allograft. They will take a part of the nerve and they will try to graft it from their own body. See, I know there are some topics which you can't get into it, but please be quiet. There are few who are listening to it. Yeah. And second is regeneration of this nerve. Once the axonal injury or nerve injury happens, the regeneration and repair takes years, from 1 year to 10 years also. It is not a sudden process. And the peripheral nerve injury, that is the sciatic or axonal injury, is the commonest injury which everybody is getting. And motor coordination is poor. Once they got nerve injured, they can't coordinate walk properly and seek lifelong support. And when this axonal injury will be cured by itself is, if two transected ends remain close at a close proximity. This end and this end can be at the close proximity, so that the front part will grow and come and touch. It's not like bone which will grow in both the sides and fuse. Only the front part will try to fuse it. So it has to be in close proximity and shares all the morphological hallmarks of nephropathy. So this is a very serious condition, and the only treatment option is allograft. Is the only option, and it can't be cured all of the sudden. It takes years. This is a question now. So what we did is, so we know why we chose biological nerve graft. So we are searching for tissue which looks similar to that of the nerve in our body. So the umbilical cord, so it has it can act as a small nerve once you cut, and we can tie it together. And this umbilical cord has a region called as Vatanjali, which has a lot of stem cells in it. And they have shown that this stem cell can be regenerated to neurons. So that was the aim. So we thought, is there any tissue inside our body which can be taken and it can be sutured, and we found that the umbilical cord, and it has very less immunorejection also. And what are the set as an absolute criteria? As I said, whenever we do a research, we will set an absolute criteria. must be a biological tissue, because we speak about pharmaceutical, 3D printing, all these things. But reproducibility, nanoparticles, all these things are difficult. That architecture, that anatomy, making that anatomy is very, very difficult. So, must be a biological tissue. It has to be taken from the body. So that the architecture need to be maintained. Is point 1. And it need to be less immunogenic. Since we are taking from the umbilical cord, if it is put in anybody's body, it should not be immune rejected. It should not be eaten by the macrophages. And it need to be less immunogenic. And must have the capability to suture with nerves. Suturing the nerve is a very, very complicated process. If you have a anatomy and physiology, you can go and look at the structure of the anatomical structure of the nerve. Suturing is a very, very technical process. So that tissue has to get sutured easily. And second thing is that tissue need to be rich in growth factors which can trigger or regenerate axon, and preferably an allograft. It should not be taken from the same person, it has to be taken from somebody and has to be put. So when we look into that, we found in the body there is something called a umbilical cord which possesses all these properties. It's less immunogenic, can be trimmed and sutured, it's an allograft, and it is rich in stem cells, and it can differentiate into neurons, and rich in growth factors that can differentiate also. Okay, so that umbilical cord was our target. And what we did is, first what we did is we have to check whether it can able to, that umbilical cord has stem cells, what they have said, whether it can differentiate into neurons or not. That is very important, no? So for that what we did is we collected bone marrow, umbilical cord, and then we tried to passage it. These are little bit technical things, and we tried to find out whether it can able to differentiate or not. And here you can see, so finally when we use retinoic acid, it is differentiating or conditioning media, so that whatever stem cells which we collected from Vatanjali is differentiating into neurons. So conceptually we are right. Now let us go and operate. Let us trim. This is a umbilical cord which has one artery and two veins. The between region is called as Vatanjali, which has rich in stem cell. So we cut a piece of sciatic nerve or the axonal nerve, the peripheral nerve, and we sutured with the umbilical cord. And some we did reverse suturing, some without suturing we put, and then we made some walk track analysis, and we found that the umbilical cord is working good. Without treatment or with reverse treatment, this umbilical cord is guiding the neuron to go and make connection with the cut region of the axon. So, but yes, it's fine. What we can do now, we submitted this in a transfusion conference in San Francisco. We went to USA and we submitted, and out of that submission, so you need to go for conferences abroad. That's actually the marketplace where you can sell your product, where you can also find new opportunities, and they will also come to our hospital and invest. They came to our hospital and they have invested more than 100 crores for their project. So but what is the translational value of this of doing all this nerve graft? So we found, when we isolate the nerve graft out of it, we found some infiltration of the immune cells inside. So it is not completely immune free, it is immunogenic. So so that whenever we give a graft, we need to give a immunosuppressant, and it needs a intense large animal experiment is warranted. So these are the limitations. We didn't stop with this one. So from the limitations only we will find an opportunity. So how to overcome this immune filtration is the next question. So what we did is, so we applied the similar strategy for cardiovascular disease. So how we plan to reduce the immune rejection is, we removed all the cells from the matrix. For example, we took allografts, xenografts like bovine pericardium or jugular vein, and we removed all the cells from that. Only the matrix will be there. We cross-linked with nanofibers, and then cross-link that under UV. We have some different process that work is also patented, so that it will work. So now the immunorejection is free. And if you want to treat, you can treat. And all the product, these are the product which are in phase 3 clinical trial. Mostly it will end in next year, and all the 3 products will come to the Indian market, and this is going to be the indigenous graft from India, because we used to purchase from abroad which cost 40,000. Now it can be given with 20,000 only. And the work is very simple. Only thing is, when we are looking at certain, for example, when we stop with this nerve itself, we can't think about this, how we can overcome the immune filtration. And since it's a cardiothoracic center, we have cardiothoracic surgeons where they want graft. So these are few of the products which are processed things, bovine pericardium, jugular vein, all these things. And next, I want to tell about the failure also. That is also very important. No? We want to develop artificial blood. So artificial blood, since we have stem cells, these hematopoietic stem cells can produce RBCs. Okay, we try to do 2D culture and we found it's producing very good amount of blood, and the hemoglobin content and everything is good. Now we plan to build a prototype bioreactor, and it was funded by DRDO and DST also. But both the things, when we try to build a bioreactor and we use the same bone, like decellularized bone, and then we nano coated it, on that we seeded the hematopoietic stem cells, and we added media, and we try to generate RBCs. So it's like I am spending 10 rupees and I am getting 50 paise. How it could be? So it's a big failure. So after that, we didn't look into it. And these are some of the other clinical trials, that is stem cell therapy. These are the therapies which are currently going in the hospital. This is also in phase 3 clinical trial. So there are something called as terminally ill patients, terminally ill heart cardiac failure patients, which means that only replacement is an option because their ejection fraction will be less than 40. So if they get heart, that's fine. Otherwise, if you are able to prolong or procrastinate their death by 6 or 1 year by using the stem cell therapy, then that is good. By the meantime, if they get a heart, it's good. So for end stage heart failure, we are using the stem cell. It's not actually the stem cell, it's a mononucleated cells which have stem cells in it. And also this amniotic membrane, the placenta, the outermost cover, we removed all the cells and we put on burn, 3-degree burn, and it was curing like anything, and it is transforming into skin also. So these are few of the products which are in the clinical trial. And there are few success stories and then few failure stories. And since, so first I spoke about how to make an absolute criteria and a relative criteria. For that you need to be very clear about what is the problem and what are the key markers I want to measure, for that what I should use. So now I'll speak about the current work which we are doing, and we are probably going for US patent now is, we plan to do a CAM-KK2 inhibitor, because this is an emerging target, that CAM-KK2 call model and CAM-KK2. So there is no inhibitor till in the investigational phase, which means that in clinical trial also there is no drug. And there are few limitations with the existing experimental CAM-KK2 inhibitors in terms of selectivity, all these things. So what we thought is, since it's being an ML and AA era, we implemented AA and ML in this project. How we did, I'll tell you. So first it's a normal process. First we did the FDA approved drugs, we dropped in it. In the top 10 we took, top 10 based upon the rank and interactions, we took the top 10. And next is where the ML or the machine learning come into picture, that we converted the top 10 into step 1 analogs by using star drops or bioisosters. We made an analog, and the most important thing is those analog can be synthesized in 1 or 2 steps. Okay, so from 10 compounds we can able to generate nearly 2000 analogs. We try to dock all those 2000 analogs. from the top 10 analog 1, we try to generate analog 2. So our aim is the interaction need to be maintained, and the docking score need to be good. So like that we found out a compound, and it can be synthesized in 2-3 steps, and it need to have a unique structural features, then only I can go for patent. So this is the picture which is at the down bottom. I didn't show big. It's out of machine learning approach. This is called the STSNE, or cluster analysis, which we perform so that we can able to figure out which cluster we have to take. So what it will do is, it will break each chemical structure into 512 bit vector maps. Like 512 dimensions it used to screen. We have 2-dimensional and 3-dimensional. Here it is 512 dimensional. It used to screen the chemical molecules, and it will show which cluster we can take. Likewise, we chose a cluster, and this is a molecule. I didn't name the molecule. This is a molecule, and you can see we did a in silico dynamic study for 250 nanosecond, and you can see that the stability of the complex, the stability is within 3 Armstrong. That is, the protein is within 3 Armstrong, and if you look at the ligand, it is in 1.5 Armstrong, which means that the complex is very more stable, and it retain all the important interaction, and we are able to synthesize, and that is showing very good activity with CAM-KK2. So now we are planning for in vivo study. So with this short duration of time, I can able to give you a little bit what we did, but these are my ancillary domains. So I want to end up with this quote whenever I study my human anatomy book, and then that pathology book by Robinson. So I used to say the same. This quote will come into my mind till I am reading it. Understanding the basic is easy. Appreciating the nuances takes years. Once you started appreciating the nuances, research is easy, innovation is easy, and it can be translated anything. And you should always keep in mind the things which is already known, and the things which is already known, and the things which is not having a translational value need to be published. The things which have a translational value need to be patented or to be taken further. Got it? So with this, thank you. If you have any questions, you can ask me. Yes, thank you.
Any questions from the participants? If you have any questions, please do ask. Any questions?
Thank you, sir, for guiding us the aspects of drug identification related to clinical data, its selectivity, industrial base, risk-benefit ratios, and its advancement for the better health status of the human community. Now, I would like to request Dr. Sanjay Shirsagar, sir, to kindly summarize the session. Please, sir.
Good evening. First of all, I must congratulate Dr. M.S. Gosavi, College of Pharmaceutical Science, for organizing this wonderful session. I thank Dr. Amrutkar sir for inviting me and addressing the chair for this session, as well as Dr. Pingre sir, co-convener, Dr. Shedke madam, for inviting me for this session. I must thanks to Dr. Jaiknath for addressing such important topic, which is always close to our mind, how the drug are being discovered, and as a pharmacist we must know how the drug come into the market, because every lay person expect from us that how the drugs exactly come into the market, and sir in very simplified way explain how it is comes into the market. But the final product which we see, it took a large efforts to come into the market, as been quoted by him, that almost 1,000 molecule need to be worked on to have one successful molecule, and it take almost 10 to 12 years for a drug to come into the market with a cost of billion rupees for a one successful marketed drug. So you can imagine the kind of efforts which has been put for the research work, efforts which has been put for the successfully completion of a project, and the patience the scientists have for the completion of this project. I must appreciate the sir for addressing the selection and the validation of the process, as well as the way he explained the things with a case study. And I must appreciate the sir that it took courage to discuss regarding the failure project. And the project he tried with, that is a need of hour, that is red blood cell RBCs. There are scientists who are working since long time on this project, and they are still waiting to have a success. And sir have put his efforts for the completion of this project, as well as he explained us the successful project of the stem cell therapy, and it given us the hopes that one successful product will be helpful to the society, as well as it will give us a chance to become a billionaire. So with this hope of with the hopes, the research work he has presented, I have once again expressed my sincere thanks to sir for this wonderful session, and MS Gosavi Institute College of Pharmaceutical Science for organizing this seminar. Thank you very much.
Thank you so much, sir. Now I would like to request Dr. Sanjay Kshisagar sir to please felicitate our resource person, Dr. Jaikant Chandrasekharan sir, with a certificate and memento as a token of gratitude. Please, sir. Thank you, sir. Now I would like to request Dr. Jaikant Chandrasekharan sir to felicitate our session chair, Dr. Sanjay Kshisagar sir, with a certificate and memento. Please, sir. Thank you, sir. Hereby, I conclude that the last session is over. Thank you, everyone. I request Dr. Shilpa Harak ma'am to conclude the day 1 of International Conference.
Good evening. I'm sure you are all waiting. Thank you, ma'am. All impatiently waiting for going home. I'll just summarize in 2 minutes, and then you can have your certificates as well as tea. So in the morning we begin with the inaugural program, which was presided by Honorable Sir Dr. M.S. Gosavi sir, with our chief guest Dr. Kohli ma'am, and yes, please be with me for the last 3 to 4 minutes of the day. Please be with me. So after that we had the first plenary session, for whom we had the speaker Dr. Kanchan Kohli from Lloyd College of Research and Management, Noida. She spoke upon the paradigm shift from conventional to innovative pharmaceutical research. The session was chaired by Dr. Mrudula Bae from MVP's College of Pharmacy. The second session was conducted by Dr. Vidhu Airy ma'am from Jamia Hamdard Institute, Delhi. She spoke upon phytopharmaceuticals, a holistic approach to plant-based drugs. The session was chaired by Dr. Rupali Patil from KK College of Pharmacy, Nasik. The third session, which just concluded, was based upon well thought out and sought out strategies in drug discovery by Dr. Jaikant Chandrasekharan from NIMS, Hyderabad. and the session was chaired by Dr. Sanjay Shirsagar sir from Met Institute of Pharmacy, Nasik. After this, we will be having the conference certificate distribution. So please be patient. What we have done, we have segregated the certificates college-wise. So we will call out a name of the college. You can come up, and one person from that college will come and collect it. Okay, and after that you will have tea, which is in the adjacent room only. So please be patient. I call upon Deepali Bandari ma'am